All right, it looks like that the majority of the attendees have loaded into the system. So I just want to welcome you guys to the webinar hosted by AOCS, brought to you by Novozymes. I'm Matt Thompson. I'm the North American Regional Marketing Manager here at Novozymes. And the topic of today, we'll be talking about the introduction of a phospholipase in the environment of an alkaline refining and how we've demonstrated increased yield with that product. So with that, some folks may not be familiar with Novozymes. So I want to give a quick introduction to Novozymes. There is a slight delay in the slides as they load. But Novozymes is a biotechnology company, and we utilize biotechnology to solve problems of the world. So we have hundreds of molecules that we've launched since 2000, and we have 19 sites in R&D sites in North America or in the world with 1,600 employees, and that's really our how. So our how is we leverage nature to improve the world, but it's really our why that has us here. As my system loads, so we have some really ambitious targets at Novozymes around sustainability. Sustainability is really who we are. So both in the food, the fuel, and the water space, we're looking to really improve the reduction of CO2, the amount of food waste that takes place, and the water used in different processes. Oh, excuse me. So with me we have Mark and Per Munk. Both are major players here in Novozymes for in our VOP space. Mark has over 6 years, and he's supported the oils and fats space in various range of roles, ranging from very technical to now commercial. Welcome to Mark. And then Per Munk has over 30 years of designing and introducing enzymatic technologies in oils and fats applications. And he's led the development work for this webinar. And welcome to Per.
Thank you.
Before we get started, I'm going to explain a little bit of the format. I'll engage Mark and Per on a various number of questions, building on the article in January from Inform Magazine. The title of that magazine or article was Phospholipase for Enzyme-Assisted Alkaline Refining. If you don't have that article, you'll still be able to follow along in the discussion, but we encourage you to get a copy. Also consider becoming an AOCS member. The annual meeting will be held May 3rd to the 14th. That's going to be a virtual meeting, and we hope you join us there. There's also going to be someone monitoring the chat for questions, so feel free to submit questions throughout the webinar, and at the end we'll review those questions and try to get to you. If we don't get to you during the webinar, then we'll get back to you after the webinar because we can see who posted that. So Mark, why don't you give us a 30,000-foot view of the VOP process or vegetable oil processing, and then we can zoom in on some of the specifics.
Sure, thanks Matt. It's great to be here with everyone. To get started, I'd like to give a high-level overview of where enzymes get utilized within vegetable oil processing. Today, enzymes can be used across the oils and fats value chain and applications all the way from extraction to late-stage modification. On the front end, enzymes can be used to improve palm oil extraction. And in the late stage, enzyme can find many uses, including production of specialty fats, biodiesel, and oleochemicals. For example, enzymatic interesterification is used to create specialty products without creating trans fats and without using harsh chemicals. In refining and degumming, producers use enzymes to break down the phospholipids in order to increase oil yields, and in this webinar, we will dive much deeper into that topic.
So in this webinar, we're going to focus on the refining aspect of the process. And I have some slides to illustrate some potential pathways. And Mark, if you can just take us through those potential pathways and how the refining step is used in different plants. And
sure.
Load here.
Yeah, we'll let the slide load in. In vegetable refining, we typically talk about two pathways: chemical refining and physical refining. We sometimes refer to chemical refining as caustic or alkaline refining because in this process, caustic is used to remove fatty acids as soaps. This caustic treatment, also called alkali neutralization or saponification, removes fatty acids as well as phospholipids and other impurities into a side stream called soap stock. This process can produce a relatively high yield loss due to oil trapped in the soap stock. An alternative to chemical refining is physical refining, which avoids the use of caustic and the formation of soaps. In physical refining, fatty acids are removed by stripping in the deodorization step, and phospholipids are removed in a degumming step. This results in two byproduct streams, a gum stream and a deodorizer distillate stream. In addition, producers can use a simple water washing or water degumming step to remove the bulk of phospholipids. This process only removes the so-called hydratable phospholipids. This is done in many cases to reduce the phosphorus content prior to transport, for producing lecithin, or as a pretreatment for subsequent chemical or physical refining.
Great, Mark. So we have the physical refining, we have the chemical refining, sometimes referred to as alkaline refining, then we have our water degumming. So why don't you take us through how enzymes have contributed to these processes?
So enzymes have found benefit for use in degumming for more than 20 years and also installed in more than 40 plants around the world. Phospholipase enzymes are successfully used to break down the phospholipids or gums, making them easier to remove and improving the process yield. There are also different types of degumming enzymes which have been developed, and we continue to develop new and improved ones. In physical refining, PLA-type degumming enzyme is often used for deep degumming, and this enzyme releases fatty acids from the gums, thereby improving the solubility and viscosity of the gums. This effect is to reduce the gum volume and increase the neutral oil and fatty acid yield. It also helps to get a more consistently low phosphorus content out of the degumming step. In water degumming, a PLC-type degumming enzyme is sometimes used, and this enzyme releases diglycerides from the gums, which also reduces the amount of oil lost in the gums. The effect is to reduce the gum volume and increase neutral oil yield. For chemical refining, this has been done without the use of enzymes historically, and the yield losses of oil into soap stock can be relatively high. That is, until now, where Per and his team have developed a new process which combines enzymes with the traditional chemical refining process to give higher yields than ever before.
So thanks for that, Mark. So we're talking about introducing enzymes into various different processes. We introduced Novozymes, but some folks may not be familiar with enzymes. So Per, why don't you give us an idea? Although enzymes aren't necessarily novel, they are novel to this process as far as the chemical refining process. To help us understand a bit about enzymes.
Yeah, okay, thanks. Very basically, enzymes are proteins that catalyze biochemical reactions. They are everywhere in our body, in plants and in microbes. There are many different types of enzymes, but those we are using today is the types called hydrolytic enzymes, breaking down some components, and that's what we need for the subject of today. Novozymes produces the enzymes which we have found in nature, and we move the genes into a production organism, fermented in large fermenters, harvested and sell it to our customers. But besides finding the new enzymes, we also work a lot with developing new processes for our customers and to help to make more sustainable processes and so on. And this is very much based on the properties of the enzyme. You have to treat them gentle to make them active or keep them active. So the conditions are very mild conditions with temperature and pH and so. And we use them in the processes for giving a more sustainable process to substitute some of the more harsh processes typically.
Thanks, Per. So with that, you kind of compared the enzymes to the chemical alternatives, and it seems like that the enzymes are a lot more specific in their approach. So I guess the enzymes themselves are quite specific, and you mentioned PLC and PLA, and maybe you can expand a bit on what those terms actually mean and how they differ.
Sure, it's important to distinguish between the different type of enzyme and what they're doing. We have a slide here showing picture of phospholipids. The phospholipids is the molecules consisting of two fatty acids, a glycerol backbone and a phosphate group with a side group here, the side group which is called X. This side group can be different compounds. It can be just a hydrogen, it can be a choline group, it can be ethanolamine, inositol, and then a few other minor things around. But these groups also help us giving the phospholipids a name. It's important to distinguish the phospholipids. So with the X, it's a PA, phosphatidic acid. With a C, it's a PC, phosphatidylcholine. With an E, it's or ethanolamine, it's a PE. And then we have the PI for the phosphatidylinositol. So these are the phospholipids we work on. And we have different enzymes doing that, doing some jobs there. And Mark told about the PLAs for hydrolyzing off one of the fatty acids. There are two types of PLAs. One taking the fatty acid in the first position. This is called a PLA1. And likewise, the PLA2 take the fatty acid off in the second position. And then today we are talking about a PLC, which is hydrolyzing between the glycerol backbone and the phosphate group. And that produces a diglyceride. Another important thing here is that when we mix a bit of water, a couple of percent into oil, the phospholipids tends to collect on the interface between the oil phase and the aqueous phase. So we create an emulsion to get the reaction going. And the material is locating itself at the interface, and we have the fatty acid compounds sitting closer to the oil phase and the phosphate group closer to the aqueous phase. And when we have the hydrolysis going, you see the PLAs produce a free fatty acids, which will go into the oil phase, and the PLC makes the diglycerides, also going into the oil phase. It's quite obvious that you get more oil mass, so to speak, into the oil phase when you use a PLC. So that's the big advantage here. So that's how it works, how we split the phospholipids and get them to create more yield.
Thanks for that, Per. So we've covered, both the process options and the enzymatic approaches. What does implementation look like? And Mark, I'll turn this over to you to help us understand, how do you incorporate the two?
Yeah, thanks, Matt. Here I would like to make a kind of a quick comparison of the traditional alkaline refining process on the top picture and the enzyme-assisted alkaline process using Quara Boost. Quara Boost is the name of our new enzyme for this application. As we have touched on, sodium hydroxide is added in the traditional alkaline refining process, converting the fatty acids to soaps. Those soaps and gums are then removed via centrifuge or soap stock. Sometimes acid is added to improve the removal of phospholipids, and sometimes the process is preceded by a water degumming step. Now, in the second picture on the bottom, you see the new concept of enzyme-assisted alkaline refining. And for this new concept, we use one of our new enzymes, Quara Boost, which is a proprietary blend of PLC enzymes, and we integrate that into an alkaline refining process. We do this in a relatively simple way, where we add an enzyme retention tank along with temperature and pH adjustment in the same general ways that we do for physical refining with enzymes today. In this way, the enzyme will generate extra yield in the form of diglyceride and neutral oil, while reducing the gum volume without major changes to the existing process flow.
All right, Mark, so you took us through the comparison, the alkaline and the assisted alkaline with the enzyme. Now you went through the implementation. Let's maybe go a little bit more detail on that.
Yeah, so basically in the enzyme-assisted alkaline refining, we can break this down into 4 basic steps. There's acid addition, pH adjustment, enzyme reaction, and saponification or neutralization. The first step and the last step are really same basic steps you have in the traditional chemical process. The middle 2 steps are the ones where we're modifying a bit for enzyme addition. But walking through the steps, in acid addition, we of course add in acid like phosphoric acid, and this helps to chelate metals and convert phospholipids into the more hydratable forms. The acid dose is then suggested based on the quality of the oil. then the next step, pH adjustment, is done particularly for enzyme to get the proper pH for the enzyme to work most efficiently. So this is a quite important step. Here we add the amount of sodium hydroxide that's needed to obtain roughly a neutral pH for the enzyme to work most efficiently. And it is key to get the pH and temperature in the proper range always for the enzyme to work efficiently. Then once the pH and temperature adjusted, then the oil is going into the enzyme reaction step. We first dose in some water, 2 to 3%, and adjust temperature to around 55 degrees C. A relatively small amount of enzymes, such as 150 part per million, sometimes can be less or more, but it's added, and then the reaction takes place for a couple of hours. Also, that can vary depending on how you have the plant reactors designed. After the reaction, then it goes to the remaining, the final remaining caustic added to saponification of the fatty acids, and this is then added in the amount to neutralize all the acidic species plus a small surplus. That is the total amount of caustic added. We have a calculator to support getting all of the proper calculations of the dosing rates. After this, the stream is heated and centrifuged as in the normal process to separate the soap stock.
Thanks a lot, Mark. So we compared the chemical refining process, the enzyme-assisted, and how to implement. Now, Per, we're going to turn it over to you a bit to help explain some of the benefits that we've seen from this implementation.
Yes, and the benefit, it all comes down to yield and extra yield. So I'm going to talk to you about where does it come from. We're back to the phospholipids, the composition of phospholipids. You see the bar chart in the middle. It's showing an example of a composition of phosphor compounds in the oil, which is phospholipids, the PC, PE, PI, and PA. Then there are some lysophospholipids also, and some other phosphor compounds. And what you do in the factories, many people have an ICP analytical equipment where they measure the phosphorus compounds, the total phosphor, and that counts these elements I just mentioned: phospholipids, lysophospholipids, and acids, and so on. We are in our lab have been more interested in the full, you could say, the intact phospholipids, which we measure by our NMR equipment, and that is not, we're not counting the lysophospholipids or the acids, because that's not anything that our phospholipases are working on. For this phospholipase C is also not hydrolyzing a PA, the phosphatidic acid, due to the difference in the group sitting on the phosphor. So that's we have to leave out in gaining the yield. So we follow also the reaction by measuring the content of diacylglycerols, the DAGs, which are formed from hydrolysis of the PC, PE, and PI. And we normally say we get at least 75% conversion hydrolysis of these three compounds. And then it's important to say that when we hydrolyze these phospholipids, they will also lose their ability to emulsify the neutral oil. And that means we also get a release of neutral oil coming into the oil phase along with the diglyceride formed from the reaction. Yeah, and if we look at the next slide, we have, sorry.
That's a good point, Per. I want to highlight on that is that, you know, we have the DAG release into that's being contributing to the mass of oil. We also have more oil being released due to the improvement of the emulsion.
Yeah, and then we have tried to make an easy to understand answer to how much yield will you get, and that is on the slide here. Is the graph with the two lines. When people ask us what can I expect, we can ask them what is your phosphorus number by your ICP analysis, and then we can explain that has 1,000 ppm of phosphorus in the oil. You can look at this graph and see you will probably be creating at least 1.2% of diacylglycerol, read from the blue curve. and we can translate that into a yield of at least 1.75% of total yield. So this is a handy little graph to look at when you want to have a good guess on what you can expect on the yield. We can calculate it in more details by an Excel calculator as shown below the graph, where we take the different things into condition, which I list here on the left. You have your ICP data, which shows other compounds than just what we are looking for, but we can estimate. It shows probably. The phospholipids is only 80% of what the ICP shows, and we also have to subtract what we expect is the PA part of the composition, and typically the PA in an oil is between 5 and 30%, and we very often see 10% of PA in the composition, so that's also calculated. And then we expect the release of oil, neutral oil, being somewhere between 25 and 75% of the DAC formed. That we can also add in this graph here. We added 80%. But one thing we don't count here is actually the elimination of a separation step, which was, like Mark told you before, you sometimes have a separation to get the phospholipids out. Of course, you want them in to have the full yield possibility, but also by having just a centrifugation, one less centrifugation, you will have extra yield just from that. That's not in our equation here, so I would call this these estimates quite conservative in any event. So.
Yeah, we know that these are just estimates, and based on the lab data and the information, also some of the other industrial trials we've been doing. But what are some of the parameters or process variations, Per, that could contribute or alter the results that we're seeing here?
Yeah, there are several steps. Of course, this acid pretreatment, the acid chelation, which is done to hydrate the phospholipids for better performance of the total process. It's done today already in the alkaline process. It has to be done in a good manner to get access to the phospholipids to be hydrolyzed. And then we of course have to make sure that the enzyme is having a good time, and that is on pH and temperature, and that has to be controlled, of course. The dosage, I'll come back to that in a moment, but you can see what you can do with dosage. You can adjust that and optimize that. And then we are adding a bit of water for the reaction to take place. We're talking 2.5 to 3% total water in the reactor. And then we of course have to be correct on the total sodium hydroxide content to get all the free fatty acids saponified as it should be.
Great, Per, thanks for that. So what we're going to do now is just dig into some of the performance data that we've seen.
Yeah, we've chosen to select some data from our trials with showing a rapeseed oil and a soybean oil, how it behaves to the reaction. I will again start looking at the phospholipids, and here we have showed two curves on an enzyme reaction, Quara Boost reaction at 50 degrees Celsius for 60 minutes. And we see how we start out in rapeseed oil. It has a total of a bit more than 1,000 ppm phosphorus measured by our NMR, so it's a bit high in ICP if you at that number, but we're only looking at the phospholipids here, and you see how the PC is the majority. We have PI, PE, and PA, and then we have made an experiment where we dose different levels of Quara Boost, and for the rapeseed oil, you can see the more enzyme you dose, the more hydrolysis of the phospholipids you get. And as I mentioned earlier, the PA is not touched at all by Phospholipase C. Unfortunately, we have not found any Phospholipase C that can cope with PA, and it seems to not be around anywhere. The other compounds are hydrolyzed. PI is a bit hesitant on conversion, we must admit, and that is seen in some cases, but it is hydrolyzed to some extent. If we look at the soybean curve, the situation is quite different. here to the right. It's not containing so much phospholipids, but still quite a lot to work with. And we see the same tendency. The PC and the PE is dropping, but here in the soybean oil, it is dropping very fast. And we reach, you could say, an equilibrium at already 100 ppm doses of Quara Boost. So in this oil, in this case, it would not pay off to add more than 100 ppm if you have everything in full control here.
Thanks, Per. With that, quick question for you. One thing I wanted to highlight is to ensure that the audience sees that we do have different phospholipid contents between the two oils. We're talking about over 1,000 here and just under 500 in the soybean. But you see the PC pair and the PE dropping significantly faster in the soybean. Do you have a hypothesis on why that may be? Is it because it's starting with more to begin with?
It's related to the structure of the phospholipids and to what enzymes we have. We have a mix of Phospholipase C, and they are very fast on the two the PC and PE. That's kind of the nature of it, you may say. It's every component is reacted in different reaction rates.
Thanks, Per.
Okay, if we look a bit deeper into how we translate to yield, I will talk about this using one number of for estimating yield again, which is DAGs, the deacylglycerols. And here we have reactions with rapeseed oil and soybean oil in two graphs. The first graph is now saying, telling about the reaction time in minutes with doses of 150 ppm Quara Boost at 50 degrees Celsius. So that's the doses chosen for this setup. And what you see again here is that the soybean oil, the black curve, is quickly reaching, you could say, an equilibrium already after 60 minutes. Not much extra happens at that dosage of enzyme, whereas the rapeseed oil still is moving upwards even after 2 hours of reaction. You should also notice I've earlier said that you will get at least 75% of your PC, PE, and PI hydrolyzed, but here you can see we are quickly with the soybean, we are quickly above the 75. We actually get close to 90, or a bit above 90. And still a little bit above 80% for the rapeseed oil. So these are the DAG numbers. We can see how much percent of maximum we will reach. You can turn it around and say, well, instead of changing the reaction time, we can change the enzyme dosage. It's looking at the same data, you could say in another way. We have the doses of Quara Boost on the x-axis, and you see again here that the soybean is quickly reaching the plateau, more or less a plateau, where we still are working on the rapeseed oil for a longer period. It's very much related to the difference here to the difference in phospholipid content in the oil, the rapeseed being more than the double. So there is more than the double work to do, so it of course takes a longer time. But that can then be adjusted by calculating what is paying off here.
So Mark, I'll turn this over to you a bit. This is Per, thank you very much for this lab information. Can you help us? And that's a good indicator of the performance, but what have we seen out in the field?
Yeah, so of course this is a new solution, and the reason we're kind of introducing this through the webinar, we're still, as Per's kind of showing here with all the data, we're still learning all the time. We do have commercial trials that are running or planned to be running very soon, and across different regions. And so far, we've seen quite positive results. And we also have a patent pending on the process, and just looking forward to explore this further with different different regions and different oils and so on.
Great. So how would some members of our audience, we've showed some calculators and showed some lab information. How could they value the process in their own plants, and how can they understand how this solution can maybe impact them?
Yeah, so we have a few things. We have tools. We have a calculation tool, for example, which allows you to estimate the value of this process compared to other processes. And so we're now developing and improving on these kind of tools, and we have them available for ourselves and for customers to work with. You can run simulations in these tools to determine the impact of various parameters, like Per is talking about, different phosphorus contents and so on, and how that impacts the economics. Also, we discuss processes, in depth with customers and run tests in our lab on various oil types, just similar to data Per is showing here, to really find the best solutions for each situation. I think that's one thing that's consistent, is that it's, I guess, not consistent in the sense of, there are differences, in different oils from different places, and maybe different season to season and so on. So it's still important to, to get in this lab data and combine that with these models.
Yeah, that's a good point. There's a little bit of a progression there. So we do have a calculator, and if the audience members would like to utilize that calculator and run some scenarios, our contact information will be available at the end of the presentation. So please reach out to us, and that gives us a pretty good understanding of what, say, potentially could and the value could be created given the modeling, but also getting in touch with us with running samples from your own plants and your own setups. We do know that the variability is pretty vast. It can be, both from oil quality, and then of course that will define the result, which is why Per kind of outlined some of the considerations on how those can take place. But concluding the presentation, then I think it's important just to highlight where we are right now. And we've had a PLA in the market for the physical refining for some time. We've introduced a solution in water degumming, and now we're introducing a solution in the alkaline or physical refining. So it's a really exciting time for us, and we'll enter the Q&A portion of the webinar. So we did have someone monitoring the questions, and we'll go through there. And of course, it's an open conversation here with Per and Mark if there's anything we would like to highlight in the webinar.
I actually have one thing I didn't get a chance to tell or to emphasize. We have talked about some variations in the process, which make you think what would be the variation if I install this process with the enzyme. I think what is fair to say is that we are in a very nice situation with this enzyme process, because we have a very robust process. You are having at the back end your normal saponification, where you neutralize the oil, where you actually clean it up, everything. So you can have the situation that for some reason the enzyme had a bad day, a low dosage or whatever is fluctuating. You will have the cleanup by the saponification. Maybe you won't get the optimal yield, but the oil quality comes out still very good. So I think it's a very important point that we have a very robust process with this enzyme application. Thanks.
Thanks, Per. So I'll go through some of the questions now, and Mark and Per and also myself, depending on what the question is, we'll try to answer them. They are coming in, so the system is filtering them as we go. But one of the questions that comes up has to do with the increase in DAG, and if there are any considerations or requirements given that for mitigations against GE and fatty acid esters. Per, do you have an answer to that?
Yeah, I can answer that. Yes, it's correct. That is a concern which has been around for several years now. It came from discovering years ago, and it was found that the DAG content, especially in palm oil, was responsible for the formation of these compounds. And it was also found that which took place in high temperature treatment in the deodorizers. Of course, we also have deodorizer in soybean oil, rapeseed oil, and so on. But we don't have the need for high temperature and so on. And in the alkaline refining system, we don't have free fatty acids to remove. So with the DAG content we create here, which is not at all in the range we see in palm oil, it has been found that it cannot contribute to these compounds.
Thanks, Per. What is expected? What is the expected? It says TFA of the resultant soap stock. That's total fatty acid, I guess would be the question. So what is the expected? Is there a fatty acid change in the soap stock at the backside of the plant, Per?
The soap stock is changed while we are picking out some of the phospholipids, but we are not producing free fatty acids in this process. The enzyme we use is producing only diglycerides,
Right.
so we're actually not doing anything about that. If you have more or less free fatty acids in the soap stock, that will be because you are not saponifying enough. But I mean, that you normally do. So you have the same amount of fatty acids coming out in the soap stock as you normally do. The concentration is a bit different because you have less phospholipids.
Mm-hmm. Yeah, and I guess you have more yield out of it as well. So the concentration, I guess, would be different.
Yeah.
There was a question about equipment. So it seems like there was a question around, does the size of the reactor or geometry of the reactor drive the reaction in any way? Does it increase or could it limit the creation of the yield or the DAG? Is there any equipment requirements we would be recommending?
Yeah, I can answer to that also. We have for many years been operating degumming, enzymatic degumming, and what seems to be very good is these compartmentized reactors where you have a constant flow through, for instance, 4 compartments. And then we are talking about holding time, a total holding time in this 4 compartment tank of 1 to 2 hours, and that is you say something you calculate with the engineering and what does it cost to size the tank for 2 hours. It costs a bit more, but maybe you then can save a bit on the enzyme. I should of course not say that. I would rather have you making a small tank and buy some more enzyme, but that's the reality.
Mark, maybe you want to comment to the materials the tank should be made with.
Well, I was just going to make the comment that you know, with these type of cases, of course, we're you know, deeply involved with the enzymes and also the process when it comes to details around the equipment design. Then we, you know, work with a number of different engineering partners closely together, and you know, they get involved obviously in designing the details around the equipment. But another thing is what we see on this kind of a process too, where you're kind of doing maybe a retrofit from, say, an output process to, okay, I want to add enzyme. Then, you know, sometimes you can find out ways to use existing tanks modified to use as an enzyme reactor, or in other cases, you may, you know, design from scratch through the engineering company to design it proper, like Per mentioned, with compartments and so on. So we see at least you know, in our experience with enzyme degumming, you know, there's a lot of different configurations out there. There's some, like Per said, some of the more kind of engineered design, multi-stage reactor with compartments and so on, and other times it's maybe some more simplified, modified reactors. And so then you can work with different situations, whether you know, within certain budget, and also you know, just depending on you know, your enzyme dosage as well. A more efficient reactor, you can you know, save on the enzyme or save on the amount of volume you need.
Thanks for that, guys. A lot of questions coming in around process, so it's interesting. So pH adjustment, for example, are there any specific tools or instruments to incorporate? So it sounds like that maybe folks aren't pH adjusting in certain cases today. So can you give us just a quick on that, Mark? Maybe you're the best guy for that one.
Yeah, I think, yeah, as I was mentioning earlier, was just around the pH adjustment. So with this process, yeah, we do need to add that pH adjustment there for the enzyme. And so, one way is you can more or less use some, lab experience to say, roughly how much caustic you need to add and then scale that up into your plant. But then also you can, we have methods, just basically what we call pH water extract method. So you just extract some sample and contact with water and measure the pH of that water. And as I mentioned, in this case, we need something around the neutral pH. So those are the kind of checks you can make on the system to make sure that you have the right pH for the enzyme. And typically once you have the process in place, it's not like you're constantly adjusting that up and down all the time. You generally some plants really very rarely change the caustic on something like that.
Yeah, so once it's installed and implemented and running, then it's not something that you have to play with a lot. The enzyme does have tolerance within a certain range, so it's pretty robust as a process. It's not something you need to really keep the closest of eyes on.
Yeah, it doesn't have to be super exact to a certain pH. You have a certain window that you can work with here.
And if you think of the alkaline refining or with the caustic additions, do we see reduced caustic additions with the use of the enzyme? So is there, let's say, an impact of some of the other additives that are being used now? Do we see those increasing or decreasing after the enzyme is introduced?
We see almost the same, but a little bit more caustic is required to neutralize hydrolysis products from the hydrolysis. So the phosphate groups, they are producing a little bit of acidity, but that's very minor. But you take it into account and add that the back end, and that's it. But it's not exactly the same. It's a little bit more.
There's a question about quantifying the gum removed. What's the easiest way to separate the gums? I mean, I guess we're talking about centrifugation. I guess maybe Per, if you can mention in the lab, how are we doing that? And then Mark, maybe what do we see out in the field?
Quantifying the gums, yeah, the way we do it is about like centrifuge. You actually have a picture on the screen. That's what we do in the lab to show and convince ourselves it works. This is the soap stock from the process you're seeing here. So that's what we do in the lab. We are working with other process controls for trying to set up instrumentation, near instrument to follow the soap stock composition. We're talking to near suppliers to try to create a partnership with a producer who want to do the soap stock or do this process with the enzyme. We would like very much to include also a near instrumentation to be able to follow it real time, and that will enable the producers to optimize the enzyme dosage and the other parameters in the process to get the most out of it. So yeah, centrifugation is the easy one, and in the longer run, we probably are talking in near instrumentation if it pays off, of course.
Yeah, we're excited about that one for sure. Mark, anything to add on that one?
No, I was just saying the plant. I think I've seen just with the load cells and so on, you can measure the weight collected on those gums if you want to try to mass balance.
Great. There's several questions around different feedstocks. We went over soybean and we went over rapeseed. So maybe some comments on corn oil and palm.
Yeah, for corn oil, yeah, the same principles apply, but there's one thing on corn oil that often comes with a very high free fatty acids, and you perhaps rather wanted to refine that in a physical refining, else you turn a lot of You turn all the free fatty acids into soap. So that is the problem with corn oil. There's plenty of phospholipids, so in principle you can do the job, but you have a lot of soap formed from the free fatty acids. And in the other end of the scale, you could say palm oil, it only has very little phospholipids, corresponding to around 50 ppm of phosphorus, and that will hardly pay off to buy enzyme to recover yield from these only 50 ppm.
Yeah, so palm maybe not the best fit, and it sounds like what you're saying around the corn oil is that although this exact process that we're going on over this webinar may not be the right fit, there are solutions, and we do have some ways to reduce the phosphorus content and generate more yield in corn oil, just maybe not with the caustic or alkaline process.
Yeah, then we're talking physical refining, and that's also possible, as you say, sure.
Yep. I think we covered some of these. People are asking about lower quality oils or maybe niche oils, low margin markets. Is enzyme degumming compatible with these low margin markets, such as edible oils, or only with specialty oils? Okay, so that's actually a good question. So is this only with specialty oils, or have we seen this, or is this solution feasible for edible oils?
I think the entrance is if you are refining on alkaline refining here, then you are in principle capable of doing this insert of the enzyme step. Then you have to find out, does it pay off? And that depends on how much phospholipid there is available to hydrolyze. You have to pay for the enzyme, you have to pay for the retrofit or whatever. So it depends on how much phospholipid you have. And I would say if you are below 300, 250 ppm phosphorus, it's probably not paying off to think in this direction. But above, you should consider it.
Yeah, and I just want to add to that the question seems to be framed in a way that it's targeting towards specialty oils, but I would say we're actually targeting to more broad applications. So the way that we are trying to come up with the solution is in a broad application, such as edible oils, biodiesel pretreatments, and other types of applications. It's not low volume niche specialty oils. It's not the only target area for this. I'm just going through the questions to see what else. This is a good question because I think it's relevant. So there was questions about results from a commercial scale use. And I think it's fair to say, and I'd like you guys to build on it, but it's fair to say that we've seen the results in the lab are translating to the results in the field is the way I would say it. And Per and Mark, you want to comment on that?
Yeah, well, from my side, I would just say, yeah, right now it's still in the early stage. So we have some, let's say some early stage data, and everything is looking good. But you know, that's what we're kind of focused on right now, is having more plants up and running and getting more, even better handle on specifically the performance across a more wide variety of situations.
Yeah, it is a relatively new application, so we're still trying to collect as much data as we can and get as much implementation as we can, but everything's pointing in the right directions. There's a question around how much water we need and how much oil is lost through the soap stock, it looks like, is the question, compared to the alkaline refining process. So is there a significant amount of water that we need to add? And is there basically more or less oil lost through soap stock?
There's not a significant amount of extra water. We can do with 2.5% of water around the enzyme step. And that's you also need water when you do the saponification and so on. So you can just calculate that amount, and you can balance with that. So you don't need total extra water. And yes, there's less oil in the soap stock because we gain that as yield. So so that's really the point of it. So the soap stock is changing with with respect to composition, less oil, and difference in the phospholipids. We also see a lower viscosity in the soap stocks, but we don't have much data on the soap stock. We are working on that to characterize that better.
There's some questions around the P content in neutral oil. Let's see, can you comment on the P content in neutral after the primary neutralization separator? So I guess it's a pretty specific question depending on how the plant is set up, because I think it's also in line with maybe some comments around if you did water degumming, and if we have any data or experience with introducing this process to water degummed oil.
That sounds like a water degumming application, or what?
Yeah, I think that. I mean, I think that the rough answer to that is, you know, fundamentally the product works with phospholipids. So the higher the phospholipid content, then the more yield and the more performance we would expect out of the addition to this process. So if you're taking a significant cut of the phosphorus out through a water degumming process, there's still you know, 200 plus ppm potentially in there to go after. But of course, the expected result is going to be significantly lower than it would be if you had 1,000 or 1,500, you know, ppm oil that you were using from the baseline. So it's to say that the economics would have to pan out. We'd have to have that conversation to figure out where the value is, if it makes sense. But we would expect that the overall solution would have a less value than higher phosphorus oils. Still have a few more questions coming in. I'm trying to get through them all. What is the impact on the bleaching earth consumption? Does this have an impact on the bleaching earth process or the bleaching steps and downstream, guys?
I'm not sure about that. I don't see why it should impact the.
I wouldn't think.
Yeah, I was thinking through it. I mean, we wouldn't estimate that there'd be any impact to the downstream based on the enzymatic step that's been inserted. So I'd say maybe we haven't tested or looked at that specifically, but there's no hypothesis for us to think that that would be the case.
Right.
There's still questions that relate to different feedstocks like sunflower and rice bran. And I think the question for that, or at least the answer to that, guys, is I think it's also in line with understanding the phospholipid content of those oils. So it kind of goes backward to it. So whether it's a soybean oil or rapeseed oil or corn oil or whatever type of vegetable oil we're talking about, it's really the phospholipid content that drives a bit. And as Per showed earlier, the gross phospholipid content is interesting, but also the makeup of those phospholipids. I don't think we'll go into the makeup of the different substrates on this call, but it's something that we could have conversations about if you just reach out to us. Sometimes enzyme rates improve the introduction of small amounts of KOH. I'm not familiar with that. Per, do you have any reaction to that question?
Not really. I mean, we're all the time emphasizing the importance of pH should be correct. Whether you use sodium hydroxide or potassium hydroxide, it's not really a question. It's maybe easier to work with potassium, but also more expensive. But it is actually the pH as such that is the important part.
And with that, I think I'll go through the questions, and we have most of the questions that we think are the most relevant for this conversation. And then the questions that maybe we should take offline, we'll have a conversation with those folks who have reached out. So with that, I think we're any closing words from Per or Mark, and then we'll close up the webinar.
Yeah, I would like to thank for the opportunity to discuss this in more details. I think it's a nice concept and it has been widely accepted for the folks who have presented it. So it's looking good, and it's been an interesting development over the last couple of years. So thank you for your time.
Yeah, I would just like to add to that, just to thank everyone in the audience and the opportunity here, and also thank Per and his team for, all the years of work going into this, and now we're kind of on the other end. As a production guy myself, is anxious to get this out into more plants, and so if you are interested to learn more, we're definitely here, available and ready to discuss.
Yeah, and for me, it's very exciting to be a part of this, given that we've been in the space from a physical refining. We entered the water degumming, and now we're in the chemical or alkaline refining stage. So we're trying to introduce our solutions to every process that we're aware of in order to increase its yield across the industry. We do see that feedstocks are going to continue to be in high demand, and we see a really strong market in this area. So we actually encourage you to reach out to us. Our emails are on the screen. Get us through LinkedIn or any other ways of contacting us, and we're happy to collaborate with you if you have a plant that would like to investigate this solution, or if you have other ideas that maybe we should be looking into that we haven't presented. So with that, I want to thank everybody for their time, and we'll be closing down the webinar shortly.